this. Right. So today we are going into chapter 11, which is muscular tissue. Last week we were talking about the various muscles. Today we're going to look at the structure of the muscles. So we talked about some wrappings of the muscle in the lab. So in the last, probably some of you have already done that lab, and you know that the basic unit, that is this little tiny little thing which you see here, that is a muscle fiber. Okay, so this is this tiny red dot here, and you can see many dots here. That's a muscle fiber, and a muscle fiber is actually a muscle cell. And around each muscle fiber, there is a wrapping. And that wrapping has a name, and that wrapping is called the endomysium or endomysium. Now, there are bundles of muscle fibers. You can see this is a bundle. And this bundle is called a fascicle. And there's a wrapping around the fascicle. And that wrapping is called the perimysium. And you can put all these fascicles together. So it's like it becomes a big entity, and that is what you call the whole muscle. And that is surrounded by another membrane. So there's a lot of wrapping. There's a first wrapping, second wrapping, third wrapping, lots of wrappings, okay? And all these wrappings are of importance. It's not a waste at all. The reason why we have a lot of wrappings is because the muscle needs to be attached to the bone, and all these wrappings come out and they form what is known as the tendon, and the tendon attaches to the bone. So that's the purpose of all these wrappings. So it's a very strong system that you can see in the muscles. So that's the structure of the muscle. This is a muscle, this is a tendon, and that's the bone. Let's have a look at each muscle fiber. So let me go back. So I'm talking about this little thing here, this little dot. That's a muscle fiber or a muscle cell. Okay, a muscle cell is called a muscle fiber. I want you to write that down. A muscle cell is called a muscle fiber. Okay. Now, just like any other cell, when you think about a cell, you always think about a round thing, but over here, it's this is how the muscle cell is. We call it a muscle fiber. And like any other cell, there's an outer membrane called the sarcolemma. So anything with muscle, we add the word sarco, S-A-R-C-O. The cytoplasm is called sarcoplasm. The membrane or the plasma membrane is called the sarcolemma. And muscle has got something very unique. Inside the muscle fiber or muscle cell, you can see some cylindrical structures. You don't see this in any other cell. These cylindrical structures are called myofibrils. So myofibril, they are cylindrical structures inside the cell. Okay, so the cell is packed with these myofibrils or fibrils. And these myofibrils, they run the entire length of the muscle fiber. And it is this which causes the contraction of the muscle. Okay, so it is a myofibril or fibril which causes the contraction. Okay, so let's have a look at the structure of that myofibril. So here you can see this is a myofibril, okay, and you can see that surrounding the myofibril there's a little wrapping, and that wrapping is called the sarcoplasmic reticulum. You have heard about the endoplasmic reticulum, right? In a cell, that's one of the structures in a cell, endoplasmic reticulum, right? In the muscle, there is a sarcoplasmic reticulum. And where is that? That is surrounding or wrapping the myofibril or fibril. Okay? So why do we need this sarcoplasmic reticulum? For the okay, it's a wrapping, but there's another purpose. The purpose, what is that ion which muscle needs to contract? Muscle needs something to contract. No, ATP is a power source. It needs ATP, yes, but there's a ion which is needed for contraction. What does muscle need? You should know that. Calcium, yes. Without calcium, muscles don't contract. So calcium is needed for muscle contraction. Okay. And where is the calcium stored? Where is the depot for calcium? Yes, it is that's the reservoir. But in the muscle, calcium has to be released, and it comes from the sarcoplasmic reticulum. And where is the sarcoplasmic reticulum? Powering the myofibrils. Okay, you get the structure? Okay, it's not complicated, but you have to get it right. So here you can see myofibril, and it is surrounded by this wrapping. So this is actually the sarcoplasmic reticulum. And in the sarcoplasmic reticulum, there are some enlarged areas. You can see an enlarged area here, another one here. They are called the terminal cisterns. And the purpose of this is to hold the calcium. So if you look at the structure, next to the terminal cistern, which is the enlarged sarcoplasmic reticulum, there is a tube-like structure which is called the T-tubule. Actually, that is actually an extension of the sarcolemma. So you can see it's like a tube, and it's on either side you can see the terminal cisterns, which are the enlarged areas of the sarcoplasmic reticulum, and the three together, that is two terminal cistern and a T-tubule together, is called a triad. Okay, so terminal cistern is where the calcium is stored. Muscle needs calcium for contraction, right? And that calcium is stored in the terminal cistern. Clear so far? Okay. Now, let's have a look at the structure of the myofibril. Myofibril is made of smaller units called sarcomere. That's the model that I showed you in the lab, in the last lab. Or people who have done that lab, you would have seen that there was a model on the table, and that model had a red and blue, and it was, it was sliding. Yeah, that's the model we are talking about. That model is a model of a sarcomere. So the sarcomere is the structural unit of a myofibril, and it is actually, there are multiple sarcomeres. So there's one here, It repeats, another one here, another one here. It extends through the whole length of the myofibril. So what is a sarcomere? I told you it's a functional unit. It is an area between two lines or two discs called the Z disc. So there's a C line here. There's a C line here. This area between the two C lines is called a sarcomere. And inside, you can see there's a thin filament and a thick filament. The thin filament is called actin. Thick filament is called myosin. And these filaments slide. So when they slide, the muscle, the length of the sarcomere shortens. So the Z lines come together and the length of the sarcomere is shortened. And that's when muscle contraction happens. So you must imagine there are thousands of sarcomeres, which contains the actin and myosin filaments or the thin and thick filaments. They are sliding. When thousands of that slide, there is a movement happening, and that's what we call muscle contraction. Let's have a look into the mechanism, what's really happening. So I told you it slides. So this sliding is called the sliding filament theory. So the basic theory of muscle contraction is the sliding filament theory. So let's have a look into the structure. So the thick filaments here, this is a thick filament. That it's made of a protein. These are all proteins. So this thick filament is made of a protein called myosin. The thin filament is made of another protein. You can see the thin filament here, which is red in color, thick is purple. And the thin filament is made of a protein called actin. And I told you they slide. That's why we call it the sliding filament theory. So how does this sliding happen? So in the thin filament, so this is a thin filament here, you can see that it's made of a protein called actin, which is a main protein, the main structural protein, but there's two other proteins there. One is called the troponin, and the other one is called the tropomyosin. They are regulatory proteins. They decide whether to slide or not. So when a muscle is relaxed or when there's no contraction happening, the regulatory protein, which is the tropomyosin, there are two, troponin and tropomyosin. The tropomyosin covers the binding sites. So the actin and myosin, they have to bind to each other in order for that sliding to happen. So normally, when a muscle is not contracting, the binding sites are covered by the tropomyosin. So something has to happen to make that cover move. That covering has to be, the tropomyosin cover has to be removed. And that is, the key for that is calcium. So that's why we said calcium is very important for muscle contraction. So what happens here when there is calcium, it's like a key. The troponin suddenly gets activated. The troponin moves the tropomyosin out of the way. Now the actin and myosin, they can bind. The actin has got binding sites. So let's go back to the myosin. The myosin has got a head. This head attaches to the binding site of the actin. So we're going back here. And when the calcium binds to the troponin, troponin lifts the tropomyosin and the actin binding sites are exposed. The myosin will grab the actin filament, and this grabbing, when it attaches and grabs it and moves it, this is called a cross bridge. So a cross bridge is formed that pushes the filament, and that's what we call the sliding filament theory. Clear? Is everybody with me? Okay. You have to know this mechanism. Okay. So moving on, stop me if you don't understand any part of it. Okay. So when you look at a muscle under a microscope, what you see is this. You can see dark areas, you can see light areas. So what do you call this? Striations, yeah. This is a skeletal muscle. Skeletal muscle is striated. That's a feature of skeletal muscle. And the dark regions are called the A band. So this is the A band, and it is dark because it contains thick and thin filaments. But the other region, which is a light band, is light because it only contains the thin filament. Is there any other muscle which is striated other than the skeletal muscle? Cardiac, yeah. Cardiac muscle is striated, but what's the difference? One is you control it. So what do you call that? It's the cardiac is, it's not under your control, but the skeletal muscle is under your control. Okay, so next we are going into we're going to talk about the brain and all those things, and that's when we are going to talk about how impulses start in the brain and what happens and all those things. We leave that for the time being. What about the smooth muscle? Do they have striations? No, that's why they are called smooth muscles. Okay, where are they seen? Where's the smooth muscle seen? Blood vessels in the internal organs, yeah, internal organ or the viscera. That's, say it again. In the colon, that's a digestive tract. That's all smooth muscles. That's not under your control. It's involuntary. Digestion happens whether you like it or not. You cannot make it happen. So that's why it's an involuntary muscle. So skeletal muscles are voluntary. You can decide when to contract it, which means it is controlled by nerves, and these nerves come from the brain or the spinal cord. So what are the nerves which carry the signal from the brain to the muscle? There's a name for it. Motor neuron, motor. It's a motor neuron. Is there any other types of neuron? There is a sensory neuron. Say you're feeling cold. How does the brain know you're cold? Because you have receptors in your skin. Those receptors are connected to nerves. They are called the sensory nerves. They take the impulses from the skin to the brain. So there's two signals flowing. One signal is flowing from the skin to the brain. The other one is flowing from the brain to the muscles. Two directions. And we'll be talking about all that when we are talking about when we are in the nervous system. So if a motor, so this word is important, motor nerve, okay? Motor neuron or motor nerve is what supplies the muscle. What happens if that nerve is damaged? The muscle becomes paralyzed. And what happens if a muscle is paralyzed? What happens to the muscle? It shrinks, correct. What do you call that supply of a muscle by a nerve? There's a word for it. Eh? No, it's called innervation. A muscle is, when you say this muscle is innervated, that means it is supplied by this particular nerve. For example, the facial muscles, we talked about the facial muscles. What do the facial muscles do? Facial expression. Why? Why do you have facial expression? Yes, it's important for communication, but when I'm laughing, my muscles move. Why does the muscle in the face move or cause those expressions in the face? Yes, these muscles are attached to the skin, the subcutaneous layer of the skin. So when that muscle contracts, you can see, so I'm surprised now. I'm smiling. You have the sad face, you have the smiling face, you can be grinning. This is the platysma, which is acting when you're grinning. So all these things is because the muscle is attached to the skin, the deeper layers of the skin. All right. In the face, yes. If you yeah, you can, yeah, well, yeah, you can start laughing, you can stop laughing. All those things are under your voluntary control. Of course, sometimes you laugh a lot, probably. If you hear a good joke, you might continue laughing. It's because in the brain, you're still thinking about that joke, okay? So you could be laughing for a little while longer. But if you want to stop it, can you stop it? Yes, you can stop it. All right. So we talked about innervation, and then we are talking about denervation or denervation, which means the nerve supply is cut off. And when denervation happens, what is happening to the muscle? It shrinks, or the technical word is atrophy. Shrinking is atrophy. So you can write shrinking equals atrophy, okay? So a denervated muscle means the nerve supply is lost. Denervation atrophy means that muscle has started shrinking. What's the opposite of atrophy? So you went to the gym today. What's happening to your muscle now? Hypertrophy. And when a hypertrophy happens, what is happening to the muscle? Is there an increase in the muscle number of cells there? No, each cell is increasing. So actually the number of cells is constant, but each cell becomes bigger. That is hypertrophy. If there's an increase in the number of cells, there's another word for it. That's called hyperplasia. Hyperplasia. P-L-A-S-I-A. But when you go to the gym, hyperplasia doesn't happen. Hypertrophy happens. So there's an increase in the size of the muscle. No. What the brain cells does not divide. We'll come to that. There's when we are studying the brain, that's one thing. That's why if you get a spinal injury, there's no regeneration in the brain. We'll talk about that. That's a big subject. Okay, so let's look at what's happening. So from the brain, the motor neurons, remember the word motor? Motor neurons carry signals from the brain to the muscle, and when it comes here, when it's about to reach the muscle, it starts dividing into multiple branches. And this is what's called the terminal axons. So when you're studying the structure of a nerve, you can see, you learn that there's one part of it called the axon. That is what we call the nerve. And if you think of my arm as the nerve or the axon, the fingers are it's about to reach the muscle. My fingers are the terminal axons. And each terminal axon ends up in a little knob, like my fingertips are the knobs. Before it attaches to the muscle, these knobs are called the synaptic knobs. Okay, so if you draw a diagram, you should have an axon, you should have multiple terminal axons, and then you should have a little bulge at the end, which is touching the muscle. And this junction where it touches the muscle, actually it doesn't really touch the muscle. We'll talk about that in a minute. But that junction is called the neuromuscular junction, NMJ, neuromuscular junction. So here you can see this is a neuromuscular junction. And I told you it doesn't touch. There's a gap. This small gap is called the synaptic cleft. So this junction, the neuromuscular junction, is a synapsis. The nerve is going to convey something to the muscle. It's an electrical impulse which is coming, and that's going to be conveyed to the muscle, and the muscle is going to take a decision to contract. And this particular area in the muscle is called the motor end plate. It is very specialized because it contains a lot of receptors. So let's have a look at this particular structure here, the neuromuscular junction. So in the terminal axons, you can see that there are tiny little bubbles here. These little sacs, they contain an enzyme, and that enzyme is called acetylcholine. Very important, ACH, short form is ACH, acetylcholine. And they are called neurotransmitters. Okay, they are the neurotransmitters. So what happens when a nerve impulses reaches the terminal axon? These little vesicles or bubbles, they fuse with the membrane here, and the neurotransmitters are released into this cleft. Okay, these neurotransmitters, they actually attach to the receptors here on the muscle. And these receptors will actually make some changes on the surface of the muscle. So I'm going to go a little bit further so I can show you. So here you can see a signal comes here. So these vesicles, they fuse with the membrane, and the neurotransmitter is released into the synaptic cleft. This transmitter gets attached to these little receptors here. You can see the acetylcholine receptors are here on the neurolemma, that is a membrane. Let's go to the next one. Here, the acetylcholine has already attached to the receptor. What is going to happen? Something is going to happen here in this receptor. Sodium, potassium channels are going to start opening. Okay, so let's go back now. So here you can see when the acetylcholine is binding to the receptors, there's a pump in the muscle called the sodium potassium pump. Okay, this pump is acting all the time, and this pump stops. So what is this pump doing? It pumps the sodium outside, and it pumps the potassium inside. That means there's more sodium on the outside and there's more potassium on the inside. So when the sodium potassium pump stops, sodium, which is abundant on the outside, starts going back inside the cell. So normally in a cell, the cell has got a potential of, or a voltage of minus 90. Why is it minus? It's because the inside, I said, has got potassium, outside has got sodium. 3 sodiums are pushed to the outside and 2 are pushed to the inside in one go. So there's an excess of positive on the outside and a lesser amount of positive on the inside. So this is why the outside is always positive, the inside is always negative. It's a comparative thing, okay? So when you look at any muscle cell, if you put a little voltmeter on it or microvoltmeter, you can see the outside is positive, the inside is negative, and the inside is always minus 90. And this is because of the sodium potassium pump. Sodium pushed to the outside, potassium pushed to the inside. Okay, so when the impulse reaches, the sodium potassium pump stops and the sodium channels open. When it opens, sodium is going to rush into the cell. So we said the inside is negative. So when positive sodium goes inside, sodium is positive, right? Na plus, right? So when it goes inside, this minus 90 starts becoming positive. So minus 90, it becomes minus 80, minus 70, because more and more sodium is going in. Positively charged sodium is going in. So the negativity is reduced. So minus 90, then finally it reaches 0. More sodium is going in. So it becomes plus 10, plus 20. When it hits plus 30, this channel closes, sodium channel closes. Something else happens. The potassium channels open. Okay. When the potassium channels open, what's going to happen? Potassium is going to rush out of the cell. Normally, potassium is pushed into the cell by the sodium potassium pump, but here the potassium channels are open, so potassium starts rushing out of the cell. Then, when it rushes out of the cell, it starts again lowering. Then from plus 30, it goes down, it reaches back to the original resting membrane potential, which is minus 90. So these are the activities that happen inside the cell membrane. So initially, there is from minus 90, it goes to plus 30. And this is called depolarization. This particular leap upwards is called the depolarization. And when the potassium channels open and when potassium moves inside, when potassium rushes out of the cell, then the voltage will come back to minus 90, and that is called the repolarization. So you have a depolarization and a repolarization happening. Are you guys with me? Did I lose you? It's not very complicated. It's simple. If you just remember this diagram here, you will understand that this part is called the depolarization. This part is called the repolarization. On this part, you can see that the sodium channels are open, In this part, you can see the potassium channels are open. And this is all happening because a nerve impulse reached there. So this is the actual mechanism. So when a depolarization followed by a repolarization happens, we call that a action potential. So an action potential has developed. So in a muscle, an action potential develops in one part. Does it stay there? No, it keeps moving. The next part will be depolarized. The next part will be depolarized, repolarized. It keeps moving. So the nerve impulse is transmitted to the muscle. From the muscle, it keeps moving all over the muscle. So I told you, a resting membrane potential is when a cell is unstimulated. And what is the voltage inside the cell? Minus 90, correct. So the resting membrane potential is minus 90. In a nerve cell, that is slightly different. In a nerve cell, that is minus 70. I'm talking about a muscle. Muscle has got a resting membrane potential of minus 90. Okay. Nerves, you don't have to worry about it now. We're going to learn that later. That has got a different resting membrane potential. So now you understand what a resting membrane potential. And why is there a resting membrane potential? Exactly, and the outside is positive. How does that happen? Yes, there is a pump, sodium, potassium pump. Okay, they push the sodium to the outside, they push the potassium to the inside. This sodium potassium pump is what creates that resting membrane potential. Clear? Okay. Right. So we talked about action potential. We said there's a depolarization followed by a repolarization. Okay. And when this happens, this action potential is what triggers the sarcoplasmic reticulum. What's inside that sarcoplasmic reticulum? Calcium. It triggers the sarcoplasmic reticulum to release calcium. So calcium is now released. Okay. What happens when calcium is released? Yeah, muscle contraction happens how? Tropomyosin. Calcium locks into what? Troponin. Troponin moves the tropomyosin. When tropomyosin is moved, the binding sites are exposed. Actin and myosin attaches and they slide. And that's when contraction happens. Does it all make sense? Yes? No? Okay. And this is what we call contraction of muscle. So if you're with me so far, we're going into the next stage. So how does this, so I told you there's going to be a sliding happening. That sliding needs power, right? Where does the energy come from? ADP. ADP powers this process, the sliding. Okay, so here you can see this is now the same slides which we went through initially. The nerve signal comes, calcium, when it comes here, there's going to be, it stimulates these little vesicles. What's inside these vesicles? Acetylcholine. And acetylcholine attaches to the membrane here. What happens there? They open the sodium potassium pump stops, sodium channels open, The depolarization happens. This is followed by a repolarization. Action potential is created. And here you can see the sodium potassium channels have already started opening. And we said sodium flows to the inside. So there's a depolarization happening. Potassium flows out. Repolarization happening. Action potential is created. It's all repetition of what I said so far. Okay. And so an excitation from a nerve is now converted into a contraction. This is what we call an excitation contraction coupling. In the meantime, in the process, calcium is released. All these things happen, right? When calcium is released, troponin is activated. Troponin moves the tropomyosin out of the way. Binding sites are opened up. The actin and myosin, they attach to each other. The ATP moves in, powers the myosin heads. The myosin head will pull the actin. Muscle contraction happens. Does it all make sense? Yeah. Okay. Okay. So this diagram shows that already the calcium is there, troponin is activated, and the binding sites are exposed now. The myosin head is going to attach, and it pulls the actin, and this movement is called the power stroke because something is happening there. There's an active pulling, and this energy is coming from the ATP. And how does the ATP give energy to the the actin myosin to cause the contraction? Correct. ATP breaks down into ADP and phosphate. ATP is adenosine triphosphate. ADP is adenosine diphosphate. So one contains 2 phosphate, the other contains 3 phosphate. So one phosphate is released. When this is released, energy is released. So we have a power stroke, and then this attachment is called a cross bridge. And then once it is completed, then it detaches, and then another ATP comes in, it gives the energy, another power stroke happens. So a power stroke followed by a recovery stroke, another power stroke, but it has to have a constant supply of ATP. The power has to be there. What about muscle relaxation? Well, how does that happen? So when a muscle is relaxing, that means there's no more stimulation by the nerve, right? But what if there's a lot of acetylcholine there? It'll keep stimulating. So that acetylcholine has to be removed. How is it removed? By an enzyme called acetylcholine esterase. So there's an enzyme there called acetylcholine esterase, and that will break down the acetylcholine and remove it from the synaptic cleft. So the stimulation of the muscle is completely stopped. But that doesn't mean relaxation has happened, right? There is still calcium inside, and calcium will cause, it'll activate the troponin. Troponin will remove the tropomyosin. The binding happens. So that has to be removed. The calcium has to be also removed from the inside of the muscle. So where does the calcium come from? Sarcoplasmic reticulum. So it has to be pumped back into the sarcoplasmic reticulum. So there are calcium pumps there. So This is an active process. This is another process which needs energy. All this calcium has to be pumped outside, like a water pump pumping water from a flooded area. So the calcium is pumped back into the sarcoplasmic reticulum by the calcium pump, and that needs ATP. So muscle contraction is an active, power-consuming process. Muscle relaxation is also a power-consuming process. So this is what happens when the calcium is all removed, the troponin is deactivated, the tropomyosin moves back into position, blocks the active sites, and that's how relaxation happens. Just the opposite of contraction, right? Now, a muscle fiber, if it is, say for example, if a muscle fiber is stretched, would the muscle be more powerful?
No.
No, it can't be, because when it's stretched, you know, the actin and myosin will be detached. It will be like separated. There's no overlap, right? If there's an overlap, then it will start working. But when it's too stretched, it won't work. What happened if you, so is it a good idea to keep it very compressed? No, there's an ideal length. This length is called the optimum resting length, and that's between 2 to 2.5 micrometer. So this is the ideal length. If it is overly stretched, there is no overlap at all between the actin and myosin. That's not good. If it is overly contracted, then the muscle is kind of bunched up. So the myosin and actin head cannot bind effectively. So there is always an optimum resting length where in which the muscle exerts the maximum force of contraction. What happens after death? What happens to muscles? Eh? Stiffens up. Why? Why should a muscle? You're not producing any impulses. You're not thinking contract. Why does it stiffen? Stiffen means contraction, right?
You can't relax.
It can't relax. Okay. Why can't it relax? But why is it contracted?
Deteriorating the sarcoplasmic reticulum. Yeah.
Well, in that case, if it's deteriorated, it should contract. That's right. Calcium, again, calcium. So what happens? The sarcoplasmic reticulum is breaking down. So calcium is stored inside it. Once sarcoplasmic reticulum deteriorates or breaks down, calcium is released into the muscle. And what does calcium do? It causes contraction. That is what we call rigor mortis. So muscle contracts after death. But muscle cannot relax. Why? Because there's no ATP. That's where the importance of relaxation is an active process. It needs ATP. So relaxation does not happen, but eventually it will happen because the actin and myosin filaments, they will deteriorate. Eventually it will happen. But initially it will go into contraction. So that's why I'm emphasizing the importance that relaxation is an active process. Isometric contraction and isotonic contraction. So you went to the gym today, right? Okay, and when you lifted the weights, you were doing either isometric contraction or isotonic contraction. What's the difference?
Isotonic.
You were doing isotonic?
Because the muscle contracts, changes length.
It changes length. So that is isotonic. Isotonic means the muscle produces a force and there's a change in length. So what I'm doing now is isotonic. So I'm trying to pull, okay, and I'm pulling, it's coming, so the length of the muscle is shortening. That's why I'm able to pull it. So this is a isotonic contraction. What if you have a weight and you have already lifted it, which you probably did today, and then you're lowering that weight? Is the length changing? It is changing. That means isotonic can be either a shortening of the muscle or a lengthening of the muscle. So a shortening of the muscle, we call it a concentric contraction, and lengthening of the muscle, we call it a eccentric contraction.
So isometric be like yoga.
No, yoga is a complete relaxation. Yeah. So just come here. Well, actually, I can't move from here. Otherwise, I'll show you something which is very. We'll do that at the end. We'll do a little exercise at the end to show you what yoga is, okay? Well, yoga is all about relaxation, okay? And that's about it. Now let's go to isometric. Isometric is like if you're pushing against the wall. You're creating tension, but the wall is not moving. There's no change in the length. Isometric, same metrics. The length is not changing, okay? So the postural muscles, which keeps your posture. Now you think you're not doing anything, your muscles are not working. Your muscles are working. You're sitting upright. You're keeping your posture. Okay, so that is isometric contraction. What about the energy source for muscles? We said that muscles need ATP, right? It needs oxygen. Where does oxygen come from? Yeah, it comes from the cardiovascular system, okay, from the respiratory system. So basically, the oxygen will go into the lungs, the blood carries it to the muscles. So all this happens because there's oxygen being given to the muscle. But what about, where is this oxygen stored in the muscle? Does it keep coming from the blood vessels, or is there any storage areas? Where in the blood, where is oxygen? What about the blood? In the blood, is there somewhere where the oxygen is carried, stored?
Red blood cells.
In the red blood cells. And which is the particular part of the red blood cells where oxygen is stored or carried or whatever? Hemoglobin. Hemoglobin carries the oxygen in blood. In the muscle, and hemoglobin is what? It's a red pigment, right? Similar to the hemoglobin, in the muscle, there is a pigment called myoglobin. That is what carries or what stores the oxygen in the muscle. But it's a very limited supply. So if you are starting exercise, the myoglobin will release the oxygen quickly, so the muscle will have enough oxygen. But that only lasts for 10 seconds. After that, the myoglobin is exhausted. Then what happens? After 10 seconds, again, you need ATP, right? ATP has to be created or generated, and that is generated from something called creatinine phosphate. So the creatinine phosphate, this system is called the phosphagen system. So initially you have the myoglobin, then the phosphagen system. In the phosphagen system, ATP is generated from creatinine phosphate. And there's an enzyme called myokinase and creatinine kinase, CK, creatinine kinase, which transfers the phosphate from the creatinine phosphate to the ADP to create ATP. So a few words here. You have to know this phosphagen system. You have to know that it's regenerating ATP from ADP, and the phosphate is coming from the creatinine phosphate. A couple of enzymes involved. This will provide a burst of energy, but that only lasts for another 10-15 seconds. So you're up to here, up to 40 seconds now. What happens next? The third. Then something else happened. This is
start burning glycogen, right?
That's right. We are now going to go into glycogen, and that's called the glycogen lactate system. So this is the glycogen lactate system, but this is a very inefficient system because it is anaerobic. What do you mean by anaerobic? Aerobic means using oxygen. Anaerobic means not using oxygen. Not using oxygen is okay. You get energy, but you get limited amount of energy. It's an inefficient way of producing this thing, ATP. Another big problem is that when you have an anaerobic production of ATP, there's a lot of lactic acid produced. And lactic acid is what gives you that muscle pain or the burning sensation. But you have ATP produced and it keeps you going for 30 to 40 seconds. So we reached up to here, okay? Next, what happens? You're going to run for another hour. Then the cardiovascular system will kick in. Your breathing increases, your heart rate increases. It starts becoming very efficient. Initially, when you start running, it doesn't know that you're going to start running. That's why we always do warming up exercise. We're preparing the body to become efficient. So after 40 seconds, the cardiopulmonary system speeds up and the body starts delivering oxygen very fast. So just before that, we were in anaerobic respiration, which means ATP was produced without oxygen, but now we have enough oxygen because the system has now become efficient. So aerobic respiration starts kicking in. This is very efficient. And initially, the energy comes from what is stored in the muscles is something is stored in the glucose is stored in the muscle. Glycogen. Glucose is stored as glycogen. So glycogen will break down, release glucose. That glucose will give the energy. But that will last for only 30 minutes. We're going to go for an hour. What about after 30 minutes? Your glycogen supply is completely depleted. After that, fat breaks down. So the fatty acid becomes the fuel source. This is the sequence of what happens when you're doing a workout. So you start with the anaerobic, the aerobic respiration, oxygen from the myoglobin. Then the phosphagen system kicks in, and then the glycogen lactate system, which is inefficient, but because it is anaerobic. And then finally the aerobic part kicks in because the cardiovascular or cardiopulmonary system becomes efficient. Okay, clear?
You gotta wait 30 minutes before you start burning fat.
Huh?
You gotta wait 30 minutes before you start burning fat.
Yes, yeah, that's right. That's when, that after 30 minutes, then your fat starts burning, yeah. So what is muscle fatigue? That means the muscle is not able to produce any more force, tired. Why does a muscle fatigue happens? One is, we talked about the fuel source is depleted. Anything else? Metabolic byproducts, waste is produced. There's accumulation of all these waste in the muscle. Fluid is lost. Electrolytes are lost from the body because you're sweating. There's a lot of fluid loss. And muscle fatigue is actually a signal telling you, hey, I'm running out of resources now. Stop. That is what muscle fatigue is. So what happens after you've done a workout? You've done a 1 hour run. What happens after that? You're exhausted, right? Does your cardiopulmonary system suddenly stop? No. You still keep panting, and you will be consuming a lot of oxygen at that stage. Why? Because you're in an oxygen deficit stage. So this is called the oxygen debt. So we still are going to breathe heavily for a while because we have to replenish the oxygen stores in the body. ATP has to be regenerated. Lactic acid has to be converted back to glucose in the liver. Your body has to be brought back to the pre-exercise state. This is called the excess post-exercise oxygen consumption, or EPOC. So you keep breathing heavily for a while in order to make up for all the oxygen debt that you have created when you are doing that heavy exercise. Okay. Now let's have a look at the different types of muscle fibers. There are different types of muscle fibers. You have the slow twitch fibers and you have the fast twitch fibers. Slow twitch fibers is also called the red fibers, and that is what's specialized for endurance activities, which means these fibers will contract slowly, but they are more resistant to fatigue. And where do you see the slow twitch fibers? You see them in the muscles of long distance runners. That's an endurance training. Whereas in a sprinter, the muscle fibers will be the fast twitch white fibers. They contract rapidly, generates a lot of force, because if you're doing a 100-meter sprint, you have to, boom, you have to start, right? Maximum force, rapid contraction, a lot of force. But a fast twitch fiber will fatigue very quickly. You cannot keep doing that 100-meter dash for 200 meters. Because that's the type of muscle fiber which is predominant in a sprinter. So sprinters will have fast twitch fibers, and long distance runners will have more of the slow twitch fibers. Okay. What about weightlifting? We said that when you're doing weights, you're moving the muscle or contracting the muscle against a load. What happens to the muscle? Why do you go to the gym? It grows. You develop the muscles. How does it grow? By hypertrophy. So weightlifting, is it a person who does weightlifting, would he have fast twitch fibers or slow twitch fibers? Fast twitch fibers, correct.
Does it depend on the muscle?
Hmm?
Does it depend on the muscle on your calf, like slow twitch, no matter what you do?
No, when you're in a sprinting regimen, yeah. When you're in a training, your muscle will develop more of the fast twitch. But if you're in a marathon training, the muscles will develop the slow twitch fibers. These are two different fibers. Part of every muscle. You can develop more of it. That's what you do in training. So you could be made into a long distance runner, or you could be made into a sprinter by the training. What about aerobic exercises? In aerobic exercises, actually what you're doing more than the muscle development, you are developing it's an endurance training, right? You're not developing the muscles there. You are actually enhancing the function of the cardiovascular system, respiratory system, the nervous system. That's what's happening there. And a person who does endurance training will have a high proportion of slow twitch fibers. So weightlifting is fast twitch. The aerobic exercise will be slow twitch.
Make your heart bigger?
Well, the cardiopulmonary system will become more efficient. Oh, no, it doesn't. The heart, heart becoming hypertrophy is, yeah. But when you say heart is becoming bigger, yeah. Well, excessive enlargement of the heart is also dangerous, okay? So you have to, it's within limits, okay? It's when the heart, which is a separate type of muscle, cardiac muscle, when that increases in size abnormally, you call that a cardiomegaly. That's dangerous. But within a particular, whereas a skeletal muscle, you can develop it by weight training. And it can become big, and that's not a disease. Okay, that is that is to impress. So now talking about cardiac muscle and smooth muscle, is there any common, is there anything which is common in their properties? They are both, no, cardiac is not voluntary. Cardiac is involuntary. Smooth is involuntary. They both, oh, you said involuntary. Okay, yeah. Both are involuntary. What about their innervation, or what does innervation mean? Nerve supply. Innervation simply means nerve supply, okay? Yeah. So they receive the innervation from the autonomic nervous system. Where does the skeletal muscle receive the nerve supply or innervation? From the what nerves? Motor nerves. We call them somatic motor nerves. Okay. I'm preparing you for the next session, which will be the nervous system. So this is the somatic motor nerves. And what's the peculiarity of the cardiac muscle compared to a skeletal muscle? What are the similarities?
Similarity?
Yeah,
Back and relax.
yeah, any muscle does that. No, they are both striated. Skeletal muscle is striated, cardiac muscle is striated. But there's a difference there. Between the muscles, there is something called an intercalated disc. This is the classic feature when you look under the microscope. If you see an intercalated disc, that is a cardiac muscle. Why do you need an intercalated disc? To move the impulses between the muscles very fast. So if one muscle is stimulated and the other one stimulates later, if there's a delay, the heart will be, pumping in different, one part will pump early. That doesn't happen. The ventricles contract together. The atrium contracts together. It all has to happen together. And that's why you need the intercalated disc. And how does a heart pump? Where does the impulse comes from? There's a pacemaker inside the heart. It doesn't come from your brain. You don't tell your heart to beat. It beats on its own. It's called autorhythmicity. Why? Because there is a pacemaker there that produces the impulses. That impulses will move through the cardiac muscle fast. Why? Because there are intercalated discs connecting the muscle fibers. So impulses move fast, all the muscles contract together, and blood is pumped out of the heart.
What's the time?
We have a few pacemakers in the heart. You have the SA node, you have the AV node, you have, there are different levels. So yeah, there are several, yeah. But usually the SA node is called the pacemaker. That is the main pacemaker. When that fails, the AV node will take over. So there are different levels, but the SA node is what we rely on. So that is called the pacemaker of the heart. And when that fails, that's when we put an artificial pacemaker for the heart. So that's the next slide. So there's a built-in pacemaker, which creates a wave of electric excitation, and it helps the heart to contract. Whether you like it or not, it will contract. You're not controlling it, and that's called autorhythmicity. Okay, I'll be posting this. You don't have to take pictures. I'll be posting this on the YouTube. Yeah. So, what about the smooth muscle? What's the shape? What is it? Is that striated or non-striated? Non-striated. What's the shape of the muscle? It's like a spindle shaped. Skeletal muscle is multinucleated, but smooth muscle has got a single nucleus. Even cardiac muscle, single nucleus. No striations, contracts and relaxes slowly, not like skeletal muscle. It's a slow process. Where is it seen? In the digestive tract. That's what you meant by belly, right? Yeah. Yeah. So in the digestive tract, where else? Blood vessels. Why do you need smooth muscles in the blood vessels? To what? To move the blood or to control the flow of blood to a particular organ, right? To dilate. So blood vessels, so the smooth muscles can dilate, when you need more blood in an area, the smooth muscle can dilate. And when you need less, so when it dilates, more blood will be flowing to that particular organ. For example, after a heavy meal, the digestive system is active, needs more blood supply. So the smooth muscles in the blood vessels dilate, more blood supply goes there. So there's always a change in the amount of blood which is given to a particular system. So that's why, what about the respiratory system? It also contains a lot of smooth muscle. Why? No, we're talking about the lungs. Inside the lungs, there are muscles. So the bronchial muscles, they are smooth muscles. Why? You want to increase or decrease the diameter of the air passages. What about the eye? Eye contains smooth muscles. You know, there's a pupil. When there's bright light, what happens? It constricts, right? If you shine light into your eye, your pupil constricts. It becomes smaller because it doesn't want that much light entering the eye. So it reduces, the diameter is reduced. This is all done by smooth muscles. You don't do anything. It happens. But one important thing which I want you to know is peristalsis. What is peristalsis? It's what?
The way your food moves through your body.
Exactly. It is a movement of food. Does it move like, I mean, when you eat, it doesn't go there and then boom, it falls into the stomach and boom, it goes. No, it's not like that. It moves, right? How does it move? There is a wave happening. There's a wave of contraction and there's a wave, then the next part will relax. And what is really happening there, a stretch is happening. So you're eating a bolus of food. This area is stretched. When it is stretched, when a smooth muscle is stretched, it contracts. Why? Because stretching opens the calcium channels. And when calcium comes, contraction happens. So there is a calcium channels open, contraction happens, it pushes the foot to the next area. When it reaches here, this area is stretched, contracts, it pushes food to the next area. That is how food moves through the digestive tract. Okay? So this is called peristalsis. And that is a property of the digestive tract completed by the smooth muscles.
Where did it stop? Stop colon?
Hmm?
Stops at the.
So food moves through the esophagus, goes through the stomach, and small intestine, large intestine, like that. 1 second, let me just stop it. Pause recording. Stop recording.
Professor.